To Chapter 4 and General Features of Cells. This is a pretty long chapter and there's quite a bit of information, so I'm going to break it up into two sections, and this is part 1. So let's just get started with some of the beginning stuff, and then we'll move right into the different cells and their parts. So first off, the cell theory is this idea that all living things are composed of one or more cells, which we can pretty much all agree with. We know that cells are the smallest unit of a living organism, so we can have a true living organism in a single cellular form, and that new cells come from pre-existing cells. This occurs through cellular division, and that even includes gametes. In microscopy, we use microscopes to be able to see cells, so we have to magnify them. And the term magnification really is just talking about a ratio between the original size of something and the size we are actually viewing. So if 4 times the size, it's just a ratio. But in microscopy, we have two terms that you should know. One is resolution and the other is contrast. Contrast is just differences in dark versus light, and the greater the difference, maybe the sharper or more in focus something may be, or the easier it is to see. But resolution has to do with how well the microscope you're using is able to magnify the image. You can kind of think of it like a digital picture. If a digital picture is very pixelated and you used a bad camera or something didn't work well, it's kind of hard to tell if two things are two separate things because the image is very pixelated. But if we use a really good camera that has a high pixel count, then when we take that picture and blow it up to the same size, we can still see all of those differences because it's a better resolution. So microscopes, much like digital cameras, have resolution or resolving power. Now, most of the time in molecular biology, which is what this course primarily is, we would use a light microscope. And light microscopes use just regular white light, and they have a resolving power up to about 2/10 of a micrometer, which is pretty small. The electron microscope, however, is one of the most powerful microscopes on Earth, and it actually uses an electron beam. So it uses a stream of electrons instead of actual visual light. It has a resolution of about 2 nanometers, which is incredibly small. And there are even electron microscopes now that have seen gold or copper in its single atom form. So here's just a general scale. This is in your textbook. That just shows you what we can see with different types of microscopes. And with an electron microscope, you can see that we can even get down over here to this, down to individual lipid molecules. We can actually extend it down here into small molecules. And like I said, there are some extremely rare but very powerful microscopes that have gone down to even the atomic level, so less than a nanometer. Here are some different forms of microscopes, and microscopy is kind of like a toolbox. You have to use the right tool for the right job. So we're looking at the exact same cell type in all of these pictures, but each of them was taken using a different form of microscopy. So here we have a standard light microscope right up here in our top left, and this light microscope, also referred to as a bright field, this is what you're using in your laboratory. We can see the outline of our this looks like a nerve cell. Right? So we can see the outline of this, and we can kind of see the inside and the axion and all of that. So we get a general idea of the shape and morphology of the cell. We move down here and we put on a filter that uses what's known as phase contrast. Phase contrast microscopy is oftentimes used when we don't necessarily want to put stain onto a cell. And it amplifies differences, so it increases contrast, and this is one of the reasons that contrast is important. So it shows that there are actually more structures. You can kind of almost see them here in the light microscope, but here in the phase contrast, they are emphasized. So this high contrast visual here really lets us see some more parts of that same cell. Now down here in the bottom left, E, we're looking at a fluorescent microscope. In fluorescent microscopy, fluorescent dyes of different colors are attached to antibodies. The antibodies are usually specific for different parts of the cell. And when they expose the cell, we can light using using a different wavelength of light with filters. We can actually light up this cell and look at different parts of it. So fluorescent microscopy is used quite a bit, particularly in the medical fields, because we can differentiate different cell parts. You'll see a lot of fluorescent microscopy pictures throughout the semester. Now here on the right we have another standard light microscope, right? So here we have a different type of nerve cell, but with this one we have stained it. So I want you to really kind of look at the difference. Here is a non-stained cell here on the left using a light microscope, and here's a stained cell over here. And this one we can see the nucleus. We can see all of these little fibers coming off. These look like maybe some proteins of some kind. We've got all kinds of different structures that we're able to now see. Down here is what's called a DIC. And with DIC, it's a lot like the phase contrast microscope. It's usually used on unstained specimens, but what we can see here is it's almost kind of three-dimensional. So we get a little bit, just a little bit more detail. It's usually used to look at entire cells or the surface of cells, because we can really see big structures and kind of get an idea of size and shape and texture. And then down here in the bottom right, F is a confocal microscope. And confocal uses lasers. They're usually pointed to, or they illuminate different parts of the cell differently, and then they're all of these different reflections are caught on a sensor in the camera and then sent to a computer. And the computer puts all of these images together and gives kind of this three-dimensional type image that can be used. It's oftentimes, confocal microscopy is oftentimes used in combination, as is here, with fluorescent microscopy. So the dyes that are used are fluorescent dyes. They use that, the fluorescent antibodies, those sorts of things, and then they can get just a little bit more detail. Now, electron microscopes, there are two different types. There's what's known as a TEM and an SEM. Transmission electron microscopy is where we look at cross sections. And in cross sections, the electrons can pass through the cell. They are just really incredibly just a few nanometer thick cross sections of cells. They've been frozen in dry ice, or they may have been placed in some kind of acrylic. And they use a tool called a microtome to slice them into very, very, very thin slices. They're then dipped in heavy metal salts, like a gold or a copper type salt. That will strengthen the cell structures so that when they shoot the beam of electrons on them, it won't break up the cell. The heavy metals help protect the cells. Now, these electrons, if they move through the cell, then we know that's where cytoplasm would be. Think about like the cell in a cross section sitting on a plate. And then where the actual structures are, those are covered in the metals, the electrons are going to be scattered. And they're going to move all over this kind of big box that the specimen is in, that the electrons are pointing down to. And as they scatter throughout the box, they're going to do this at different angles and different speeds. And all of that information is captured by sensor plates surrounding the specimen that is being viewed. And those electron velocities and angles and such are all going to be captured. All of that data is put into software on a computer, and then on a computer screen, it uses several different algorithms to show up an electron micrograph picture. Now, scanning electron microscopy works exactly the same way, but instead of the specimen being a cross-section in scanning, we actually look at the outside. So we have transmission electron microscopy, where we can look at the inside of a cell, and then scanning electron microscopy, we look at the outside. So here we have a human egg that is being fertilized by sperm. And on the left, we have transmission electron micrograph of the human egg. And we can see there's like these little sperm out here, and then this is the membrane, and here's the inside of the egg. All of these little black dots in that are all, proteins and ribosomes, because a lot of protein function has got to go on. We've got a nucleus right over here. Now we go over here to the same egg, and over here on the right we have the outside of the egg. And you can see, look at how textured it is. It has lots of texture, and of course here are some sperm that are trying to fertilize or work their way in. They're all competing to be first. Now, in cells we have two basic types. We have prokaryotic cells and eukaryotic cells. The prokaryotic cells are the bacteria and archaea. Bacteria and archaea are not the same, but they are very similar, and they are both prokaryotes. Prokaryotic cells are complex cells. I don't want you to think of them as simple. Their structure is simple. Their morphology is simple. but functionally they are just as complex as many eukaryotic cells. They do not have membrane-bound organelles. They have a nucleus, but the nucleus is not membrane-bound. Their chromosome, or their genetic material, is exposed to their cytoplasm. The two categories of prokaryotes are the bacteria and archaea. Both are incredibly small. We have to use microscopes to see them. Bacteria are the most abundant, and majority of the bacteria found on the planet are not harmful at all. They are not considered pathogenic, but we are very affected by bacteria that are considered pathogenic, so we always think of the ones that cause disease. Archaea are prokaryotes that are much less common. They're found, I call them the X-gamers of the prokaryotic world. They're found in very extreme environments, so they could be found in lava vents under the ocean, those sorts of things. Now, your typical bacterial cell has a plasma membrane, and inside of the plasma membrane is going to be the cytoplasm. The nucleoid region, since they don't have a true nucleus, membrane-bound nucleus, it's referred to as a nucleoid region. That's just wherever in the cell its genetic material is going to be found. They have ribosomes, just like our own cells. Ribosomes are protein and RNA combined structures that are used for protein synthesis. And they also have, in addition to a chromosome, all right, so all of this blue stuff here is their chromosome. In addition to chromosomal genetic material, they also have small little pieces of DNA called plasmids. And plasmids are unique to bacteria, and they are very small, they're round, and they only contain a couple of genes. And plasmids are very important for bacteria in genetic exchange and evolution. Bacteria do have some unique external structures. They have a cell wall, which our animal cells do not. Plant cells do, but animal cells don't. They have a cell wall that they use for protection and support. Some bacteria can produce an outer coating. It's called a glycocalyx in general. Most bacteria produce a type of glycocalyx called a capsule. And the capsule is used for protection. Some bacteria use it to avoid the immune system. The ability to form capsules is considered a virulence or a type of pathogenicity in many bacteria. For example, strep throat. The bacteria that causes strep throat forms capsules. And when you get strep throat, you have severe inflammation and your throat really hurts. It's very red. All of that is because the immune system is responding to this bacteria that's causing damage, but it can't see it because the bacteria are producing a glycocalyx known as a capsule. I like to call it a Harry Potter invisibility cloak. Other appendages that may be found on bacteria are pili. Pili are used for attachment over here. They're called fimbriae, which is another term. Fimbriae are protein structures coming off of the bacteria that are strictly used for attachment, and then sometimes these fimbriae will actually hollow out, and when they become hollow, they become what are known as pili. And pili are used for attachment as well, but they're also used to exchange genetic material. Bacteria reproduce asexually. They reproduce through binary fission or cell division, which means they must exchange genetic material in another way. Since there's no sexual reproduction, they have to find other ways to exchange genetic material. And one of those ways is through a process known as conjugation, in which the pili from one bacterial cell will attach to another bacterial cell, and they will actually give the cell a copy of one of its plasmids. Over here on this particular picture, it's a little hard to see, but you can see this kind of hazy surrounding here is the capsule. This capsule here is composed of hyaluronic acid, which is a major part of our skin, and it allows this bacteria to hide from the immune system. The immune system just sees hyaluronic acid. Here is the cell wall, this really dark lining right here. That's the cell wall of the bacteria. And this bacteria right here is in the process of binary fission. And you can see what's called the septum, where the cell wall is now dividing the one cell into two. Looks like this guy is just getting ready to go into the same process over here. And this is a transmission electron micrograph. This over here is a scanning electron micrograph. So here again is just your basic structure of your cell. We have an electron micrograph here of E. coli. This is what E. coli looks like, although E. coli usually has a little more flagella. And we can see the fimbriae and pili, the cell wall, the nucleoid region, cytoplasm, and of course flagella. Not all bacteria have flagella, but some do. And those that do, they're called motile bacteria. That's what they use the flagella for. So that brings us to the eukaryotic cells. In eukaryotic cells, the genetic material is housed, and it's housed inside of a nucleus. So we have a membrane-bound nucleus, and we have other membrane-bound structures within the cell, and they are referred to as organelles. Organelles give eukaryotic cells a property known as compartmentalization. So different organelles carry out different functions, and even though the functions may be autonomous from each other, separate from each other, they all work together to make the same thing happen, and that is for the cell to function as a whole. Now, think of the cell as an office building. So each office is an individual organelle, right? Each department, and in that department you have the human resources and payroll and marketing and finance and all kinds of different operations, all of that kind of stuff. And in that office building, each of those departments is an organelle, and each department has its own area, right? Its own office area. So each organelle is a department, and then all of the proteins and functional parts inside of that organelle are the people that work in those different departments. The shape, size, how cells are organized is very different. Think about our own cells. Here on the left, I have some epithelial cells. These are cheek cells, right? The inside of a cheek, and you can see the nucleus. It's real dark right here. So we have some epithelial cells, human epithelial cells, and over here I have a human nerve cell. And what's amazing about compartmentalization and cell differentiation, it's referred to in the human body especially, is both of these cells have the exact same genome. So they're coming from the exact same genetic material, but their function, their structure, their compartmentalization is all very, very different. And they serve two very, very different purposes, but they come from the same genome. So the shape, size, organization, it's going to be different, very different between species, but it's really noticeable when we look at the difference between cells within the same organism of a multicellular organism. So here we have your basic structure of a plant cell on the left, and an animal cell on the left, and a plant cell over here on the right. And these are all of the different parts of the cell that we are going to go through, right? And down here is a transmission electron micrograph of both types of cells. There are structures found in animal cells that are not going to be found in plant cells, and structures found in plant cells that will not be found in animal cells. And we will talk about those a little bit. So first, let's go back to that idea of differentiation. In cellular differentiation, when mom's egg is fertilized by sperm, you start out as a single cell, and that cell begins to divide. And cellular division keeps occurring and occurring and occurring, and you become an accumulation of cells. But not all cells are doing the same thing. We are a multicellular organism. So our cells differentiate themselves through what's known as the proteome. So the proteome is what really determines the characteristics of our cell. And this is just a diagram known as metabolomics, and it's talking about metabolism. It's a fancy word for metabolism. So I just want to go through it real quick so you can see how it works. There's this central dogma of biology, which basically just means that everything in biology is built around this concept. It's kind of the center pillar. And it's that DNA, and our DNA makes RNA, which makes proteins. And proteins are what make other compounds. So our DNA is our genome. Our RNA is our transcriptome. When we want to send a message to the cell, the genome says, hey, we need to make a particular compound. We need to make this certain molecule. We need to make insulin, say. The gene for insulin production is activated. And when it gets activated, the gene itself is not what actually goes out and makes the protein. The gene sends out a message, and that message is sent in the form of messenger RNA. And so that's the transcriptome. So from 25,000 different genes, we can have upwards of about 100,000 different transcripts. We'll get into that in another unit. From 100,000 different transcripts, these go out and meet ribosomes, right? We've gone from DNA to RNA, and with ribosomes, we're going to make proteins. And when we make all these proteins, that's our proteome. So DNA sends the message out in the form of RNA. RNA hooks up with a ribosome, and the RNA and ribosome together make the protein that the DNA is instructing it to make. The proteins then carry out all of the cell's functions. Metabolism, although we think of metabolism strictly as metabolizing our food, metabolism is actually all of the chemical reactions that occur in a cell. So any type of chemical reaction that's occurring in a cell is part of the metabolome. And we can make thousands of different metabolic compounds from all of these different millions of different proteins. Now, what makes our cells differentiate is that what genes are expressed in the skin cell are not always the same as the genes that are expressed in a muscle cell or in a nerve cell. So even though all of our cells have the same genome, each cell selectively only expresses part of that genome. So skin cell expresses one part and a nerve cell expresses a different part. And that's what makes a skin cell and a nerve cell a nerve cell. So let's start taking a look at the inside, right? We're going to take a look at the inside of our cell. So we start out with the cytosol. That's highlighted here in yellow in our animal cell and our plant cell here. And the cytosol is the part of the eukaryotic cell that actually just kind of houses. So it's the goo inside of the cell. It's made primarily out of water. It has lots of dissolved proteins and minerals and other compounds in it. It includes, it's all of that fluid. Now, if we include everything inside of the cell, then we're talking about the cytoplasm. So the cytoplasm will include the liquid portion, the cytosol, as well as the organelles and endomembrane system that we're going to discuss. There are two primary functions that occur in the cytosol. The first is metabolism. Metabolic activities are kind of coordinated inside of the cytosol. A lot of our enzymes are found there. We initiate glycolysis, or the breakdown of glucose for energy, starts out in the cytosol. And metabolism has two different types of reactions. We have what's known as catabolism, where we break down one molecule into much smaller pieces, right? So that is used to release energy. Anabolism is when we actually need to make new molecules. So we need to repair or make a new protein for a certain function. That's an anabolic process. If we want to break down something, then that's a catabolic process. And when we get into Unit 3 in metabolism, we'll see that catabolic processes drive anabolic processes. And the production of these new molecules through anabolic processes in turn can drive other catabolic processes. So those two terms will be coming up quite a bit, and they work together. Now over on the right, the second function that's a primary function in the cytosol is translation. Translation is where we make proteins. Remember in the last slide I talked about going from the genome to the proteome. Well, that's what's happening in translation. When that mRNA molecule that the DNA sent out of the nucleus into the cytosol to meet up with a ribosome, the ribosome and mRNA together are going to translate, right? The ribosome translates the mRNA and creates a protein, synthesizes a protein. So a protein, when it's multiple amino acids, is known as a polypeptide. The information with the gene will be translated into that primary sequence that we learned about in the last unit, and primary sequence is just the sequence of amino acids. The ribosome is where actual synthesis occurs. Transfer RNA is a secondary form of RNA that's going to bring amino acids over for translation to occur. And messenger RNA, of course, is the actual information from the nucleus to create the polypeptide.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site, as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
So that is how we create the primary sequence of a protein. I'm not going to test you on the A, P, and E sites in that of a ribosome yet. That comes in another unit, but that gives you a general idea of the process. And all of this is occurring within the cytosol. It's also going to occur in another organelle known as the endoplasmic reticulum, and we will get to that as we move through the endomembrane system. So here we have another rendition of a ribosome, and you can see, I'll use my little pointer here, you can see where our tRNA is coming in. This one has this amino acid attached to it. It's going to meet up with, this is the mRNA molecule here in yellow. This is that A site, the P site, the E site, and this right here you can see is our primary sequence of our protein. As this polypeptide chain is made, in the last unit when we talked about the levels of protein folding, this is the primary structure. These guys are next to each other, so this valine and this glycine are pretty close together. They may react with each other and create hydrogen bonds, and that's where we saw that alpha helix and beta sheet. Ultimately, as this shortens up due to those repetitive structures and that the alpha helix and beta sheet formations, this brings far away regions, they coil up and they get closer. That brings us to that three-dimensional shape known as the tertiary structure. And then if we have more than one polypeptide chain, then we would move into, of course, our quaternary structure. Within the cytosol is, for structure and support, is the cytoskeleton. And a lot of times people think of the cytoskeleton as just kind of these fibers that give cells their shape. But they really have a lot of very important roles. And there are really 3 different types of fibers that make up what's known as the cytoskeleton. So the first one here on the left in that fluorescent microscopy picture up there, the green fibers are the microtubules. Microtubules are made out of a protein called tubulin, and they are hollow. These microtubules can be broken down and rebuilt so that cells can change shape. We have many cells that are able to change shape, like our macrophages, dendritic cells, many white blood cells that carry out phagocytosis and other processes. They are able to change their shape when needed. And that shape-changing ability is mostly due to the hollow nature and breakdown and rebuilding of microtubules. So primary functions of microtubules are cell shape, organization of organelles, chromosomal sorting. We find them in inside the nucleus and what's known as the nuclear matrix. The movement of intracellular compounds through motor proteins. We'll talk about those in a minute. And they are also responsible for the different proteins that make up flagella or cilia. The only flagellated cells in the human body are the sperm, so women do not have flagellated cells. And the cilia that we have are not considered true cilia. They are just cell membrane inclusions, but they include some microtubules that form them. In the second type of cytoskeletal element that we have are the actin filaments, and these are sometimes also referred to as microfilaments. They too play a role in the shape of a cell. One of their major functions is muscle contraction. You may have heard of the actin-myosin interaction. They are also responsible for cell movement and cell division, and also intracellular movement with motor proteins. Our last are the intermediate filaments, and intermediate filaments are really big in cell shape, organelle shape, if they have them, like the nuclear matrix. They are very stable. They're the most stable of all of the cytoskeletal elements. They're used for mechanical strength, so cells will stay three-dimensional and not collapse in on themselves, sometimes known as an inner matrix. They can also be found outside as what's known as the extracellular matrix, which helps to anchor our cells to each other and helps with the formation of organs. They're used in anchoring the cell membrane, the nuclear membrane. They kind of keep everything in place, and they're made out of keratins that are coiled all together. Now here we have some different types of microtubules, filaments, and actin filaments, just kind of showing you some other pictures and how they're involved. Here's that tubulin protein here, and then inside of this hollow microtubule you can see what's known as the lumen, and you can see here this cell structure, right? Here we have these filaments can be twisted and turned. These are those intermediate filaments, and they really play. You can see this matrix here. That's a really strong matrix-like structure. These are not hollow. You can see that they're quite thick, so they can be twisted. And then here are the actin filaments. They are a spiral formation of actin proteins. They're very thin, and down here they're just kind of showing you how all three of these play a role and where they're coming from. Now, one thing that's unique about microtubules is microtubules are coming from this small structure here called the centrosome. And this centrosome is, there are two of them, and the two centrosomes will actually play a really big role in mitosis, in cell division. We'll get back to those in cell division. They're not organelles, but they are structures within the cell. They're kind of where all of our microtubules begin, and they play a major function in the ability of our cells to reproduce, to divide. So that brings us to what was described with our microfilaments or actin filaments and the microtubules, and that is intracellular movement. Now, our cells have to get molecules to different places, right? And there's lots of different places. There's actually several different places they have to get things to. But not everything that is made within the cell it can necessarily just be exposed in the cytosol. We have to deliver it somewhere. So the very closely interacting with the cytoskeletal elements are three different types of proteins called motor proteins. And they are a class of cellular proteins. They use energy in the form of ATP, and they have three domains in their structure. Okay? They have the head, the hinge, and the tail. So here's the head right here. Of course, here's the hinge that's occurring down here, and then they have the tail. And this big giant ball here that it's attached to, this is a vesicle. And if the vesicle is going to come off of another organelle, this motor protein is going to attach to it and is literally going to interact with this microtubule. That's what's down here in the bottom of this picture. And the this down here is going to interact, right? So we have the tail down here. It's going to interact chemically and through energy with the microtubule and literally walk along it. So they use this walking analogy to describe what is known as dynein walking. Dynein is one of the motor proteins. So this is a dynein protein. And if you use the walking analogy, think of the ground as the filament that you're interacting with. Your leg is the head of the motor protein, and your hip is the hinge, right? So you're going to interact, and this hinge is going to bend, just like your hip can bend. And I'll show you the video in a minute. Now, when motor proteins are interacting with cytoskeletal elements, there are three different types of movement that they can carry out. They can move cargo from one location to another, like we're going to see here. They can remain in place and cause the filament to move. Think of walking in place on one of those moving sidewalks at the airport. So you go to the airport, you get on one of those moving sidewalks, walk in the other direction. So like walking on a treadmill, but you're making the treadmill move. And the last is when the motor protein tries to walk, but it's restricted in some way. It's anchored at one end or it's anchored in between and it can't move. the filament is also anchored. So instead of the protein or the filament moving, it's actually going to cause some kind of bending to occur. And this too is also going to require some kind of ATP energy. So let's take a look at this motor protein real quick. It's just a little 20-second video. But this is that dynein walking, right? And it's carrying this vesicle, and all these little nodules here on the vesicle are tags telling it where to take this package. So this motor protein with ATP is interacting. This is dynein, and it's interacting with this microfilament right here. I'm sorry, it's a microtubule. These are actin filaments. So here we have a picture, right? We have the three different types of motor proteins. We have dynein, mostly used in cilia and flagella movement, but we can also have it in that dynein walking, right? So here's these motor proteins in the dynein walking. It's our first type. Kinesin is the second type of motor protein, and kinesin is used a lot in some of the bending type motions, kind of like you're seeing down here. Kinesin does this. And myosin actually interacts with dynein and actin. So myosin is responsible mainly for muscle contraction and muscle movement. So here's our first type of movement, right? This is the movement of a motor protein along a cytoskeletal element. In this case, it's actin, and this is myosin. Myosin, right, pulls, oops, that's not working. Let me come back here. It's going to pull the actin filament this way, then it comes up and it stretches out further along the filament and then pulls the filament again. This is how your muscle contraction works. Here we have our motor proteins, right? Here we have that muscle contraction, that actin-myosin contraction. And here we have where I was talking about the filament is anchored and the motor protein itself is anchored. So instead of either one moving backwards or forwards, they just bend, and in turn they cause this guy to bend over with it. So it causes some kind of bend in the microtubule itself. So let's take a look at this quick video on muscle contraction.
Myofilaments are composed of strings of the proteins actin and myosin. An actin filament has two strands of actin molecules wrapped together. A myosin filament has many myosin proteins packed together, and each myosin protein has a globular head region that protrudes from the filament. Myofilaments can contract or shorten due to interactions between the myosin heads and the actin filaments. Contraction begins with the head of myosin molecules bound to actin on the actin filament. While still bound to actin, the myosin head flexes, pulling the actin filament along with it. This causes the actin filament to slide by the myosin filament. Then the myosin head releases from the actin and unflexes, a change that is powered by ATP. This frees the myosin head to bind with a different actin molecule farther up the actin filament. The entire process may then be repeated many times. The myosin, in effect, walks along the actin filament, moving the actin filament more each time.
So think about when you're trying to build muscle. A lot of times you take in your diet, you want to take in extra protein and that sort of thing because you want to use the building blocks of those proteins to build more of your own protein. And muscles get larger because you actually increase the number of myosin and actin filaments. And so the more myosin actin filaments you have, the more of these myosin filaments you have attaching to more actin gives greater strength. Let me try next slide here. All right. Now, other components from the cytoskeletal elements, of course, are the flagella and cilia. Flagella are much longer than cilia. They have some motility. They can be present in single form or in pairs. Cilia are very short. They usually cover the entire surface of a cell, like you can see over here with these paramecium, and they work together. But internally, they both share very much the same internal structure in eukaryotic cells. Prokaryotic cells have flagella as well. Remember the bacteria that have motility, but they use a different protein. They do not use the same protein, and their protein is actually called flagellin. The microtubules, the protein dynein and axoneme actually are what are used to make up the internal structure of flagella and cilia in eukaryotes. And the movement involves propagation of a bend. So that last form of motility we were looking at with anchoring, that's what's really going on in flagellar movement and ciliar movement. So here we have the internal structure, right? We have our of a cilia, and you can see there's some dynein here, and then we have our microtubules. There are some central microtubules. It's just a cross section, and so it causes this kind of sliding-like motion, and that's what causes these bends to occur. So these bends, kind of like a swimming motion, like a fish. So that's the end of part 1 of this lecture. In part 2, we are going to cover we're going to cover the endomembrane system and go through the actual internal organelles of the cell. So stay tuned and move on over to section 2.